Hello and welcome back to everyone. This is the third and the last lecture in this series on protein purification. So in the previous two lectures, we have discussed about the different techniques of cell lysis. That is, how can you break open a cell to release the protein of your interest from within. And then we discussed about the different methods of preparing the load. There is, once you have broken open the cell and you have released your protein of interest from the cell, how do you then clarify the lysis, that is, free it from cellular debris, genomic DNA and other contaminating agents that would interfere with further steps of purification? And how do you prepare the final load which you're going to apply onto your different beads for protein purification? So we did look in detail at the different types or different techniques of cell lysis and what are the advantages and disadvantages of each, when to select one over the other. And we also talked about ammonium sulfate precipitation and centrifugation and the different types of centrifugation, their principle, and so on. So in this particular lecture, we are going to talk about the final step, which is chromatography. So we will talk about the various purification strategies. That is, once you have broken open your cell and you have centrifuged the lysate to remove the cellular debris and you now have your protein of interest in the soluble fraction, how do you go about in isolating that protein from the rest of the lysate? So we will be talking about the various strategies that can be applied for this. So the next step in protein purification is chromatography. So let us have a basic introduction to chromatography. So what is chromatography? Chromatography is a process for separating components of a mixture. To get the process started, the mixture is dissolved in a substance which is called the mobile phase. There is a phase that is moving, which carries it through a second substance called the stationary phase, which would essentially be your beads. So chromatography was initially discovered when it was observed that when you dip a filter paper into a solution containing a mixture of different colors, you can see these colors separating out as the solution absorbs up the filter paper. And that is where chromatography has its origin from. So chroma means color. So that is where chromatography originates from. So in the previous lectures, if you have been through it, we have been talking about these beads which we are going to use to purify our protein of interest out of the solution, utilizing either some tag which is added to the protein or its physicochemical properties. So these beads actually constitute the stationary phase because they are stationary, that is, they are not moving in nature. And on the beads you add your load, which is essentially the mobile phase because it moves through the beads or the stationary phase. So the different components of the mixture travel through the stationary phase at different speeds, which cause them to separate out from one another. So what is the basis of separation? It's a difference in their migration speed. Now, this difference in migration speed can be dependent on a number of different factors, and depending on what kind of chromatography we are performing, these deciding factor actually varies. The nature of the specific mobile and stationary phases determines which substances travel more quickly or slowly and how they are separated. So as we talk more about the different chromatographic techniques, it will become clearer as to how the chromatographic columns can separate out different proteins. But for now, let it suffice to be known that you have the stationary phase, which is your beads that are packed into a column, and you have the mobile phase, which is your load or the lysate, which you have clarified. And then when you add this mobile phase onto the stationary phase, different protein components migrate at different rates Depending on what kind of chromatography you're performing, this deciding factor for this migration rate will vary, and this differential migration separates out the proteins. So these different travel times are referred to as the retention time. So longer the retention time, later the protein will elute or come out of the column or of the beads or the stationary phase. Now, chromatography can be used as an analytical tool, feeding its output into a detector that can read the contents of the mixture. So, for our intents and purposes, we are going to discuss chromatography in context of protein purification. But let it be clear that chromatography has a lot of applications in different procedures other than protein purification, like in different analytical procedures of organic chemistry and various other fields. And therefore, under such situations, the output can be coupled to a detector, which will then read the contents of the mixture that you have loaded onto the chromatographic column. It can also be used as a purification tool. That is what we are going to discuss in this lecture, separating the components of a mixture for use in other experiments or procedures. So different types of chromatography that are commonly used in protein purification include ion exchange chromatography. I'm sorry, I think there was some animation error, so number 2 came up first. Affinity chromatography, size exclusion chromatography, and hydrophobic interaction chromatography. Now, I would like to add one point at this stage, that broadly you can divide chromatography into two types. So you have your regular chromatography and you have your reverse phase chromatography. So in regular chromatography, the stationary phase is polar in nature, and so is more polar than the mobile phase. Whereas in reverse phase, the stationary phase is actually non-polar in nature. So hydrophobic interaction chromatography is a type of reverse phase chromatography, because there is a reversal of the phase from usual, where the stationary phase is more non-polar. And in case of the other three, that is in affinity chromatography, IEC and SEC, the stationary phase is more polar than the mobile phase. So that is dependent on the polarity of the mobile phase and stationary phase. You have the regular chromatography, the regular phase one, and reverse phase chromatography. So first we are going to talk about affinity chromatography. So affinity chromatography is a method of separating a biomolecule from a mixture based on a highly specific macromolecular binding interaction between the biomolecule and another substance. So as the name suggests, this chromatographic technique exploits high affinity interactions between mostly a protein and its ligand, or a protein and an antibody against it, because such interactions are of very high specificity as well as high affinity. And specificity is key to affinity chromatography. The specific type of binding interaction depends on the biomolecule of interest. So it can be an antigen-antibody interaction, it can be an enzyme and substrate interaction, it can be a receptor and ligand interaction, or it can be a protein and nucleic acid interaction. And these are the ones which are commonly exploited for isolation of various biomolecules. So essentially what is done is that your stationary phase over here will comprise of certain inert beads, say for example agarose beads, to which you have either the antibody or the substrate or the ligand covalently conjugated, so that now when you load your mobile phase onto it, the specific antigen against the antibody, or the enzyme for the substrate, or the other way around, the receptor for the ligand, and so on, will separate out. How? Because the entire mixture or mobile phase will pass through your stationary phase, but only these proteins. So for instance, if you have your antibody covalently conjugated to the stationary phase, then a specific antigen will bind to the antibody, and therefore as a result this antigen will be captured on the beads. Now the rest of the solution will flow through, and now you have your antigen bound on the beads. So this will help in separating out or isolating this antigen from the rest of the mixture. So affinity chromatography is useful for its super high selectivity and resolution of separation compared to the other chromatographic methods. Now, the other three chromatographic methods that we are going to discuss about, they basically exploit the physicochemical characteristics of the protein of interest that you want to purify. Whereas affinity chromatography exploits this high specificity and high affinity interaction. Therefore, they have very high selectivity and much better resolution compared to all the other chromatographic methods. So in a typical affinity chromatography experiment, the ligand is attached to a solid insoluble matrix which is by itself inert, usually a polymer such as agarose or polyacrylamide, and chemically modified to introduce reactive functional groups with which the ligand can react, forming stable covalent bonds. So these agarose or polyacrylamide beads are chemically modified to introduce these reactive functional groups, which can be either the antibody or the enzyme or the ligand, to which the interacting counterpart protein will bind when you pass the load through it. So the stationary phase, that is your beads conjugated or chemically or covalently conjugated to these antibodies or ligands, are loaded onto a column to which the mobile phase is then introduced. So first you take this column, you add your stationary phase, that is the beads, to it, and then you load your mobile phase onto it. The molecules that bind to the ligand will remain associated with the stationary phase, and the rest of the solution will simply flow through. A wash buffer is then typically applied to remove non-target biomolecules by disrupting their weaker interactions with the stationary phase, while the biomolecules of interest will remain bound. So it happens that when you're loading your mobile phase onto the beads, your protein of interest will bind to the column based on this specific interaction of, say for instance, the receptor with the ligand. But some other proteins also remain associated with the beads in a non-specific fashion. So because it may be so that it simply got entrapped in the spaces between the beads. So you then apply a wash buffer to remove these non-specifically associated proteins, and they get very easily removed simply by adding a wash buffer and passing this wash buffer through the beads. The target biomolecules may then be removed by applying an elution buffer, which disrupts the interactions between the bound target biomolecules and the ligand. So your elution buffer will contain certain key components that will help in disrupting the interaction between the bound biomolecule and the ligand. Say for instance, if we are doing an antibody-antigen interaction-based affinity chromatography, where you have your antibody covalently conjugated to the stationary phase and you're looking for the antigen, then what you do is that for eluting out the antigen, you simply add an excess of antigen to the solution. Now this might seem confusing to you at first, that if you are purifying an antigen, then why would you simply add excess of antigen to the solution? So let me clarify. This antigen is not your entire protein that you're trying to purify. It is simply a tag on your protein. Say for example, a very commonly used antigen tag for purification is flag tag. So you have your protein of interest and you have a flag tag attached to the protein to help in purification. Now this flag tag will bind to its antibody, that is the anti-flag antibody, which is conjugated to the beads. Now in order to elute these flag tag proteins out of the beads, you're simply going to add an excess of flag peptide. Now this flag peptide will compete out the biomolecule or the protein with the flag tag that is bound to the antibody, and it will compete out this protein to bind to the antibody, and now you will have your protein in the elution. It might seem a bit confusing, but think about it, it will get clearer. So the target molecule is thus recovered in the eluting solution. So this is a schematic depicting the principle of affinity chromatography. So what do you have? So this is your column and this is your resin. So you see these sky blue beads, it might not be very clear. So this is a resin to which you have covalently conjugated these pink molecules, which can be, say, the antibody against your affinity tag, whatever affinity tag it may be. So this is the antibody against the tag. So you have these antibodies that are covalently conjugated to the resin, and this resin makes up the stationary phase which you have loaded onto this column. So next you load your sample onto it. So your sample contains a mixture of proteins along with these green proteins of interest, and they have this yellow affinity tag which is also linked with them. Now how is it linked? Because you will be expressing your protein of interest in any target organism. So you have genetically modified the DNA such that it will express the tag along with the protein of interest. So your protein is expressed with the tag at all times. Now you load this sample onto your resin. So what happens is that unwanted proteins will pass out, and these green proteins of interest which have the affinity tag, they are bound to these pink antibodies which is specific for the affinity tag. So now you have them bound. Most of your unwanted proteins would usually pass out in the flow through during loading itself, but some may remain entrapped in the spaces in between the resins. So when you add your wash buffer, these protein molecules that are non-specifically simply entrapped within these spaces, they will also get removed. Now you add your elution buffer. So the elution buffer will contain nothing but an excess of this free affinity tag, and this affinity tag, because it will be present in excess, will displace out these green proteins from the antibodies, and it will bind to the antibody instead, and now your purified target protein will come out in the elution, and you can do further analysis or with it, or you can simply store it depending on at what stage of purification you are at. If this is your final stage of purification and you are getting pure protein out of this, you can directly go ahead for storing it. But since your elution buffer contains an excess of this affinity tag present in it, so you will have that excess affinity tag in your eluted protein solution as well. So ideally, you should remove that tag from this protein solution before you can store it or before you can use this protein for other experiments. So you can either remove that by doing other types of chromatography, or you can do dialysis to remove it. But if you have to do dialysis to remove it, you have to keep in mind what kind of what is this affinity tag that you have conjugated to your protein of interest. If Because if for instance it is a peptide, then it will be difficult to remove it by dialysis. It will not remove it very efficiently because the dialysis membrane also has its limitations and specific cutoff values and so on and so forth. What do I mean by cutoff values? That is what size of molecule can pass out or not. So you can remove a peptide tag by dialysis, but there are other methods that are more preferred. Preferably you would go for another round of chromatography to remove the tag from your protein. So next we are going to talk about the two ways in which affinity chromatography can be performed. It can be either done in the form of a batch binding or using a column directly or a hybrid mode. So what do each of these mean? Binding to the solid phase may be achieved by column chromatography, whereby the solid medium is packed onto a column. So this is exactly the same thing whose schematic we just saw in the previous slide. So you have your resin which has the affinity tag antibody against it, or yeah, let's go with antibody. So you have your resin which has the affinity tag antibody again, covalently linked to it, and you then pack this resin onto a column. So the initial mixture will first be run through the column to allow binding of your protein of interest expressing the affinity tag to the beads. A wash buffer is then run through the column to remove any non-specifically bound proteins. The elution buffer is subsequently applied to the column and the eluted protein is collected. So this is exactly what we just discussed. These steps are usually done at ambient pressure, that is under atmospheric pressure. You do not apply any external or additional pressure to it, or you do not regulate the pressure in any manner whatsoever. So basically what you have the column with the stationary phase packed onto it. You add your mixture to the column, you let the flow through pass, then you wash it to remove any proteins that are non-specifically associated with the column. And then you add the elution buffer to disrupt the interaction between the target molecule and the ligand, or the affinity tag and its antibody, and you elute out the protein of interest and you collect it. So the next way of doing it is by batch treatment. So alternatively, binding may be achieved using a batch treatment. So what do I mean by that? This is achieved by adding the initial mixture to the solid phase in a vessel and then mixing the two. So unlike in the previous method or in column chromatography where you had your resin or your beads packed onto a column, in this case in batch treatment your resin is not on a column. You have it in a vessel. Say for example you have it in a falcon tube. So you have your beads in a falcon tube and onto it you add your initial mixture. Now that you have added your initial mixture, you first allow it to mix for a certain amount of time. You typically keep it either on a tumbler or on a rocker to allow your protein of interest that is present in the mixture to bind to the beads. Now once you have done that, the solid phase is then separated from the liquid phase by centrifuging. How? So basically you have the falcon, you have the beads in the falcon. You add your protein not protein, you add your load or lysate onto the beads. You let it bind for a certain amount of time at a certain temperature. So for example, if you're doing binding at room temperature, it will be typically for a couple of hours, like say 2 or 3 hours. If you're doing it at 4 degrees centigrade, it would be overnight. So you let it mix and you let it bind. Next, you take out the falcon, you centrifuge it at a low speed, not at very high speeds, because centrifuging these beads at very high speeds, any beads for that matter, any chromatographic beads, if you centrifuge them at a speed above 3000 or 3500 rpm, that will be detrimental to your beads. So you centrifuge it at a low speed. So this will cause your beads to settle down to the bottom of your falcon tube, and on top you have the rest of the mobile phase which is not bound, and you simply remove it by decanting it. So the liquid phase is removed from the solid phase. The solid phase is then washed and protein is eluted. So how do you wash it? So once you have decanted the rest of the mobile phase from the falcon tube, leaving your beads undisturbed, you add the washing buffer to it, you mix it again, you centrifuge it, the beads settle down, you remove the excess wash buffer on top of the beads, ensuring that there is no bead loss. You repeat this a couple of times to sufficiently and completely wash the beads, and then you add the elution buffer. You allow it to mix again for some time. You allow your protein of interest to get eluted from the beads, and then you spin the falcon, the beads settle down, you collect the elution from on top of the beads. So sometimes a hybrid method can also be employed, in which you essentially allow the binding to occur in this batch fashion, and after binding you take the beads and load them onto the column and then do the rest of the steps, that is washing and elution, on the column. So hybrid method is employed such that the binding is done by batch method, but the solid phase along with the target molecule bound to it is then packed onto a column, and washing and elution are then done on the column. And this method is very commonly used in different labs. So the column method and the hybrid method are much more common, at least according to my experience, as compared to the batch treatment method. Why? Because in hybrid method, washing becomes much more easier, because you simply keep adding the wash buffer onto the column. Whereas in the batch treatment method, after every time you add the wash buffer, you have to go for a centrifugation step, and you have to then decant the excess wash buffer from the falcon, and this might sometimes lead to loss of beads which would otherwise be avoided if you wash it on the column. Now, affinity columns can be eluted by either changing salt concentrations, pH, pI, charge and ionic strength directly, or through a gradient to resolve the particles of interest. So the kind of affinity column that we took as an example was based on antigen-antibody interaction. So over there, elution involved having excess antigen in the elution buffer. But you can use either of these techniques for elution as well. So for example, you are doing a nickel NTA chromatography column. So if you're doing a nickel NTA chromatography column, then your protein of interest has a histidine tag on it. So how do you elute it? You keep imidazole in the elution buffer. And you can either elute at one step at a particular concentration of imidazole, or you can take a gradient elution where you gradually increase the level of imidazole. So this will allow for better resolution. Why? Because naturally proteins will also have some histidine residues, proteins other than your protein of interest, and by virtue of these histidine residues, these proteins will also bind onto your nickel beads. So to remove them, you have to apply lower amounts of imidazole, and at higher concentration of imidazole, your protein of interest, which has a longer histidine tag, possibly a 6 or a 8 histidine tag, that will elute out. Similarly, you can also take elution by changing the pH. For example, if you are using protein A beads for performing affinity chromatography, so protein A beads will bind to FC protein. So your protein of interest which you want to purify has an FC tag, which is, what is FC? It's the FC portion of the antibody. And protein A will simply bind to this FC portion. So to elute it out, we simply use low pH solutions. That is, we use arginine at a low pH to elute it out. So change of pH can also help in disrupting the interaction between the antigen antibody or the receptor ligand. So you can do it either directly at one concentration, or you can take a gradient, that is a gradually increasing concentration of elution. So this is a schematic representation of the batch treatment, whereby you add the mixture to batch to bind the target molecule. You remove the mixture, you add the wash buffer, you wash it and you remove the wash. You add the elution buffer and finally remove the elution buffer and collect your protein of interest in it. Now, scientists often use recombinant DNA technology to develop cells that will produce large quantities of the desired protein. So this is known as an expression system. So this is something that we have already dealt with. Like if you are purifying your protein from bacteria or from insect cells, then these bacterial cells or insect cells become your expression system. And we utilize recombinant DNA technology to ensure that this host expresses my protein of interest, which I can then purify from it. So recombinant expression allows the protein to be tagged. Why? Because you are manipulating the gene of the protein of interest. You are determining what would be the genetic sequence. So therefore, because you have a control over the genetic sequence, you can add a tag to the protein. This will therefore reduce the number of purification steps required. Why? Because if your protein of interest has a tag, you can directly go on and perform affinity chromatography. So protein tags are peptide sequences that are genetically grafted onto a recombinant protein. So often these tags are removable by chemical agents or by enzymatic means such as proteolysis or enzyme splicing. So often when you're adding a tag to your protein to help in purification, you do not want that your protein has that tag after the final step of purification. So you ensure that this tag is cleavable. How do you do that? You introduce an enzymatic cleavage site in between the protein of interest and the tag, so that once you've purified your protein using the tag by affinity chromatography, you can then add the enzyme and cleave the tag away from the protein of interest and then remove it. So now you have your tag-free protein for further studies. Now, tags are attached to the proteins for various purposes. They can be added either to the N-terminal end of the protein or to the C-terminal end of the protein. So, one thing to keep in mind is that addition of the tag should in no way interfere with proper folding of your protein of interest or with its functionality. So, the tag should in a way be inert with respect to the protein's folding and functionality. Otherwise, it defeats the entire purpose. Now, some tags can also be inserted into the coding sequence, that is within the coding sequence, and these are referred to as internal tags. But under these situations, you have to have an idea of which regions of the protein are exposed on the surface, because unless and until your tag is exposed on the surface of the protein, it does not serve your purpose. If your tag is buried within the protein, it will not bind to its interacting counterpart on the affinity column, so it will be totally useless. So in order to introduce an internal tag, you should have an idea about the protein's folding. So therefore, most tags are either N-terminal or C-terminal. Internal tags are not really commonly used in protein purification. So you have your protein of interest along with the fusion tag, and because you can design the sequence of this DNA, you can design it to include the tag. Now once you express this DNA in your expression system, which can be either bacterial, mammalian or insect, you will get your tagged protein. That is your protein with the tag. So affinity tags are appended to the proteins so that they can be purified from their crude biological source using an affinity technique. Commonly used tags include the chitin binding protein, maltose binding protein, strep tag, and glutathione S transferase or GST. The polyhistidine tag that we discussed about is another widely used protein tag. Epitope tags are short peptide sequences which are chosen because high affinity antibodies can be reliably produced in many different species. So this is what we discussed about in the first or as the first case example, where you have your high affinity, high specificity antibodies conjugated onto the resin, and you have the epitope tags that are expressed along with your protein of interest. So these are usually derived from viral genes, which explains their high immunoreactivity, and also the fact that no other protein that is expressed by your expression system will naturally have that tag because it is viral in origin. It is naturally not expressed in that expression system. So epitope tags commonly used include the alpha tag, the V5 tag, MIC tag, HA tag, spot tag, T7 tag, and any tag. It also includes the flag tag, but it is not mentioned over here. These tags are also very useful for Western blotting, immunofluorescence, and immunoprecipitation experiments. So if you go back to a couple of lectures where we discussed about immunoprecipitation, so we discussed that in immunoprecipitation, you exploit the interaction between an antibody and its specific antigen to isolate that particular protein from solution. And it is like a small scale purification procedure itself, I had mentioned. So for immunoprecipitation and for Western blotting, these epitope tags are very useful. Why? Because say for instance, you have 10 different proteins that you want to purify and all of them have the HA tag. So you can purify them all using HA beads, and you can visualize all of these proteins on a Western blot by using antibody against this HA. So it becomes very useful. These tags are particularly useful for, I'm sorry, this presentation is a bit full of errors. I don't know why this point is mentioned twice. Please forgive it. So yeah, this is what a column packed with affinity beads typically looks like. You have your, these columns are called your iconocolumns, and you have these beads, which is your resin, to which your affinity tag antibody, for example, or your interacting proteins for these specific tags are covalently conjugated. So these can be, I think, by looking at it, I would say these are nickel beads because of the blue color. And you have them loaded onto the column, and then you do the rest of the steps on this column itself. So next we are going to discuss about ion exchange chromatography. So ion chromatography or ion exchange chromatography separates ions and polar molecules based on their affinity to the ion exchanger. So it works on almost any kind of charged molecule, including large proteins, small nucleotides, and amino acids. However, ion chromatography must be done in conditions that are 1 unit away from the isoelectric point of a protein. Why? So if you remember our previous discussions where we talked about a Zwitter ion and an isoelectric point, so isoelectric point or Pi is that pH at which a protein is in its Zwitter ionic state, and a Zwitter ionic state protein has equal number of negative and positive charges. Therefore, the net charge in the Zwitter ionic state on a protein is 0. So the protein has to be at least 1 unit away from the isoelectric point. So the pH plays a very important role in ion exchange chromatography. If the pH is not right, then your protein of interest will not have the proper charge on it, and ion exchange chromatography will not work properly. So there are two types of ion exchange chromatography broadly: anion exchange and cation exchange. In cation exchange chromatography, this is used when the molecule of interest is positively charged. So in cation exchange chromatography, your resin or your bead or column by itself is negatively charged, and they are exchanging cations, that is positively charged molecules. So the molecule is positively charged because the pH of the chromatography is less than the Pi. So when the pH is less than Pi, so that means there is an excess of protons in the solution. So most of the NH2 groups will be in the NH3 plus state, which will allow the entire protein to have an overall positive charge. In this type of chromatography, the stationary phase is negatively charged and positively charged molecules are loaded to be attracted to it. And the interaction between the column and the mobile phase proteins of interest is simply electrostatic in nature. Anion exchange chromatography is just the opposite. So anion exchange chromatography is when the stationary phase is positively charged and negatively charged molecules are loaded to be attracted to it. And in order to ensure that the molecules have a net negative charge, the pH for the chromatography has to be greater than the Pi, because at a pH greater than Pi, there will be lesser free protons available in the solution. So most of the amino groups will exist in its NH2 form, and the carboxyl groups, because of the lack of protons in the solution, will release the hydrogen to form a COO minus group, and therefore the protein will have an overall net negative charge, which will allow them to interact with the positively charged stationary phase. So cation exchange chromatography is used when the desired molecules to separate are cations, and anion exchange chromatography is used when the desired molecules to separate are anions. So this might seem a bit confusing. In cation exchange chromatography, your column is negatively charged and it is exchanging cations, that is, cations are binding to it. And in anion exchange chromatography, your column is positively charged and it is interacting with anions. So the name is given with respect to the nature of the molecules interacting with the column. The bound molecules can then be eluted and collected using an eluent which contains anions and cations by running higher concentration of ions through the column or by changing the pH of the column. So simply, for instance, if you're performing cation exchange chromatography, so you have positively charged molecules bound onto the column. Now you pass an increasing gradient of salt through the column. So as the concentration of salt increases, there will be more and more free ions in the solution, both cations and anions. Now this increasing concentration of cations will displace out the bound biomolecules from the column, and you will get your protein in the elution. It will elute out. So if we take a look at this schematic, you have your cation, let's say ion exchange. You have your ion exchange column, you have the sample mixture, and this is your column, and you have the elution buffer in the charged column. So when you have the elution buffer, as you go from a low salt concentration towards a high salt concentration, gradually your proteins will begin to elute out. So for example, let us consider this was a cation exchange column. So your column is negatively charged, and all these three proteins, that is the orange, the purple and the green band, have a positive charge. So as you increase the salt concentration, you have more and more positive ions in the solution. So the green one had the least positive charge out of the three, so it eluted out at the lowest salt concentration for the three. Next, we have the purple one, which has intermediate positive charge between the orange and the green one. And the intermediate, that is the extent of positive charge on the surface of the protein, will determine how tight the interaction will be between the column and your protein of interest. More the charge on its surface, tighter will be the electrostatic interaction, and vice versa. So tighter the interaction, higher would be the salt concentration that is required to elute it out. So this orange band, for instance, has the highest amount of positive charge on its surface. So it is most tightly associated with the column and elutes out at the highest salt concentration. So if we take a more detailed look at what is happening, so this is your resin. For anion exchange chromatography, your resin is positively charged and you have these negatively charged ions associated with it. And for cation exchange chromatography is just the reverse. Your resin is negatively charged and you have these positively charged ions in solution. So in case of the anion exchange chromatography, this blue protein, which has a net negative charge, will bind to it, but the yellow one, which has a net positive charge, will be simply repelled away from the resin and it will not interact with it. Whereas in case of the cation exchange chromatography, the yellow protein will bind and the blue one will be repelled away. So once you have injected your sample, you allow the sample to absorb. You see the blue one has bound, the yellow one simply flows through, and the reverse occurs over here. The yellow one has bound and the blue one simply flows through. Next, you wash it to remove any which might be non-specifically bound. Now, how do you get non-specific interaction here? Because those proteins which have, say, lesser amount of negative charge on the surface but was simply sticking there, when you wash it, they will simply come out. Next, you desorb the sample. How? By increasing the ion concentration. So now your protein of interest, which had adsorbed onto the resin, gets removed from it and it elutes out into solution. So if we are to take a look at the various advantages of ion exchange chromatography, large volumes of sample can be loaded onto the ion exchange column. It is useful in resolving very complex samples. It is a highly efficient technique and it has low maintenance costs because you have a lot of these commercially available columns of both cation exchange and anion exchange columns commercially available, and you can load large volumes of sample onto it because there is no limit as to the amount of the sample that you can load onto it. So you can load as much sample as you want, provided that your column can bind to the total amount of protein that you're expecting to be present in the sample. You don't want that you load excess of the sample and the column is saturated and a lot of your protein of interest simply passes through because the column is saturated and therefore incapable of binding to further protein. And it is also useful when resolving complex samples. That is, you can take a lysine and you can directly load it onto an ion exchange chromatography column because your proteins will separate out on the basis of their charge. However, it has certain drawbacks as well. The protein sample to be loaded has to be at the same pH and salt concentration as the column. So it is very important to first equilibrate your column and to buffer your protein sample so that it has the same pH and salt concentration as the column, because both pH and salt concentration would play a very important role in determining protein binding to the column. It can be inconvenient for larger sample volumes since the sample has to be desalted or dialyzed after elution, because once you take the elution, you cannot store your protein in such high salt concentration. That will simply cause your protein to either precipitate out by salting out. So therefore, you have to remove that salt by either desalting or dialysis. So although you can load large volumes of protein onto ion exchange columns, this is one of the problems of it. You have to desalt larger volumes. Gradients require long equilibration because you will be taking a gradient elution where you gradually increase the salt concentration. So this requires longer times. And lot-to-lot column reproducibility is poor because these columns are commercially available and the entire capability of the column to bind and separate proteins is dependent on the resin itself, solely on the resin itself, the quality of the resin. So therefore, lot-to-lot reproducibility is not so good. It can be sometimes poor. But this is not a very major drawback because we will be using this method for analytical preparation of protein only. So this will not really make much of a difference under that scenario. The third kind of chromatography that we are going to talk about is size exclusion chromatography, which is one of the most commonly performed chromatographic techniques in laboratories. So SEC or size exclusion chromatography, also known as molecular sieve chromatography, is a chromatographic method in which molecules in solution are separated by their size and in some cases molecular weight, but most commonly in accordance with their size. So SEC is something that would typically not be the first chromatographic step that you perform when purifying a protein. It is usually the last step of protein purification, whereby once you have purified your protein to a certain extent and you have around 80 to 90% of pure protein, then you go ahead to perform size exclusion chromatography or SEC. So it is usually applied to large molecules or macromolecular complexes like proteins and industrial polymers. The chromatographic column is packed with fine porous beads which are composed of either dextrin polymers like cephadics or agarose like cephalose columns or polyacrylamide like cefacrin or biogel P. And all of these columns are commercially available. Different columns, because they are packed with different beads, they have different resolutions or resolving capacities. That is, their ability to separate out proteins on the basis of their mass varies. Some are better at separating proteins of higher molecular mass or higher size rather, whereas others are more suited at for resolving proteins of lower size. That is, they will better separate proteins of smaller size and they will not offer very good resolution for proteins of larger size, while we have other columns which will offer good resolution for proteins of larger size but not so good resolution for proteins of smaller size. So you can't really have one SEC column that can provide you good resolution for the entire range. So you have to have, or you have to select your chromatographic column accordingly depending on what is the size of your protein of interest and at what size you want best resolution. So the pore size of these beads are used to estimate the dimensions of the macromolecules. SEC works how? It works by trapping the smaller molecules in the pores of the absorbent stationary phase. So what happens is that these you have your stationary phase, right? So this stationary phase is made up of porous beads. So molecules that have a small size will enter into the pores of the beads And because they are entering within the pores, they will take longer time to make their way through the column, whereas larger molecules which cannot enter the pores of the beads will simply pass through the gaps or spaces in between the beads, and therefore they will take shorter time to pass through the column. So larger molecules, because they take shorter time to pass through the column, they will elute first. Smaller molecules, because they take this longer route, so therefore they take a longer time, and therefore they elute later when performing a size exclusion chromatography. This process is usually performed within a column, which typically consists of a hollow tube tightly packed with micron scale polymer beads containing pores of different sizes. Now, these different beads, like the cephadics or cepharos or cephacryl beads, all of them have different pore sizes, and therefore their resolution varies. So these pores may be either depressions on the surface of the beads, or they might be just channels passing straight through the bead. As the solution travels down the column, some particles enter into the pores. Larger particles cannot enter into as many pores as the smaller ones. So therefore, they migrate quicker and they have faster elution. Larger the particles, faster the elution. The larger molecules simply pass by the pores because they are too large to enter into the pores. And smaller molecules, because they enter into the pores, therefore they have a longer distance to travel, longer time of travel, and they elute later. So larger molecules therefore flow through the column more quickly than smaller molecules. That is, smaller the molecule, longer is the retention time. That is, the amount of time for which they are retained on the column is longer for smaller molecules. So this is how typically the schematic of SEC looks like. You have your porous beads. So the small tiny particles you see, they've inserted into the beads and they are sort of lost within the beads. So they are taking longer time. Whereas these medium-sized particles, they are also inserting into the beads, but they are not getting into all of these tiny convolutions. So they are migrating faster compared to these tiny ones. And the absolutely big ones, which absolutely do not enter into the beads, they migrate out the fastest. Now, for SAC, a term that is very commonly used is void volume. So what do we mean by it? So those molecules which are above a certain size, they will not enter any pores at all, and they will simply pass through the spaces, the free spaces within the column, and these molecules are said to go into the void volume, right? And for those molecules which have to undergo certain resolution, they have to enter these pores. So molecules that come out in the void volume, because they do not enter into the pores at all, so therefore there is no resolution. So whatever protein you're getting in the void volume, they are not resolved, they are unresolved. And in order for the proteins to be resolved, they have to enter at least some of the pores. Now, depending on their size, how many pores they will enter into will actually vary. So this is what a typical SEC chromatogram looks like. So this chromatogram is taken from a superose 6 column. A superose 6 column which has a 24 ml of total volume. So at 8 ml we get the void volume. So we have some protein coming out over here. There is no resolution absolutely because they are not entering into the beads. So you see at 8 you suddenly get a peak. Before that there is nothing. And then we get these different peaks for proteins that are eluting at different mLs. So therefore, this peak has a longer retention time than the proteins in this peak. Why? So proteins that are eluting out over here are smaller in size than the proteins that are eluting out over here. And this is the MAU, which is just simply the milliabsorbance, that is the UV absorbance that is measured. So coming to the advantages of size exclusion chromatography, it does not depend on temperature, pH, ionic strength and buffer composition. It simply depends on the size of the protein. More importantly, not on the size but rather on the Stokes radius of the protein. That is, two proteins of the same size might differ in their compactness. the more compact protein will have longer retention time compared to the less compact protein of the same size or same mass rather. So because SEC is independent of either of these factors, it can be carried out under any conditions. You don't have to be so careful about the pH or the salt concentration of the buffer, unlike in case of affinity chromatography or ion exchange chromatography, where the entire binding is dependent on the right pH and salt concentration. There is very little adsorption. So unlike again in affinity and ion exchange chromatography, where your molecule of or protein of interest adsorbs onto the resin of the column, in SEC adsorption is very little. There is less zonal spreading than in other techniques, and the elution volume is related to the molecular weight. Coming to the drawbacks or disadvantages, in SEC, mass is not measured as much as the hydrodynamic volume of the polymer molecules, that is the Stokes radius. So that is how much space a particular polymer molecule takes up within its when it is in solution. So like I said, proteins or two proteins of the same mass might take up different volumes of space. So that is they might have different compactness. So therefore they can be resolved out on an SEC. So SEC is not a direct measure of mass. It is a measure of the compactness of a molecule and the volume occupied by that molecule. However, the approximate molecular weight can be calculated from SEC data because the exact relationship between molecular weight and hydrodynamic volume for polystyrene can be found out. For this, polystyrene is used as a standard. So basically, when you get a new SEC column, you run a commercially available standards on it. So this standard is essentially a solution of different proteins, or it can be polystyrene, either of the two. And because from there you know what is the molecular weight of it, and you will know at what retention volume your standard is eluting out. So you can have a sort of correlation based idea of what can be the size of your protein based on the retention volume. But the relationship between the hydrodynamic volume and molecular weight is not the same for all polymers. So you can only, or for all proteins in our case, so you can only get an approximate measurement. You can never be 100% sure. Another drawback is the possibility of interaction between the stationary phase and the analyte, but that rarely happens. However, just because it rarely happens does not mean that it cannot happen. And I myself have been through an instance whereby there was some interaction between my protein of interest and the stationary phase. So once I injected my sample onto the column, I did not get any elution. So it might happen. Any interaction will lead to a later elution time, and this mimics a smaller analyte size. So because of this interaction, your protein would be eluting out later. However, you would think that because it is eluting out later, so it probably has smaller size. So it might lead to some misleading information or misleading conclusions, but it might not always be the case. And one major drawback of SEC is only a limited sample volume can be loaded onto the columns. So the typical small columns which have a 24 ml volume, over there you can load up to 200 microliters of sample, and for the larger columns which have around 120 ml volume, you can load at most 1 ml of sample. If you load more sample than that, then the resolution or the resolving power of your column is compromised, and it will not be able to separate out the proteins very well. So there is a cap on the volume that can be injected onto an SEC. So the last kind of chromatographic technique that we are going to discuss is hydrophobic interaction chromatography, which is a case example of reverse phase chromatography. So the previous three ones were all normal phase. This is reverse phase chromatography. So HIC separates molecules based on their hydrophobicity. It is a useful separation technique for purifying proteins while maintaining biological activity due to the conditions that under which it is performed, and matrices that operate under less denaturing conditions. So the principle for protein adsorption to HIC media is complementary to ion exchange and size exclusion chromatography. Sample molecules containing hydrophobic and hydrophilic regions are applied to an HIC column in a high salt buffer. The salt in the buffer reduces the solvation of the sample solutes. So we have already discussed about this in the previous lecture, where we were talking about salting in and salting out, and how differences in salt concentration in the solution can affect the solvation of the protein molecules. So at higher salt concentration, the solvation of your protein of interest is reduced, and this leads to exposure of the hydrophobic regions. So as solvation decreases, hydrophobic regions become exposed, which are then adsorbed by these HIC columns. The more hydrophobic the molecule, the less salt is needed to promote binding. So for more hydrophobic molecules, you need lesser amount of salt to bring out the hydrophobic regions and promote binding. And lesser the hydrophobicity of the molecule, greater would be the salt concentration required to promote binding. Usually, a decreasing salt gradient is used to elute the samples. Why? Because at high salt concentration, your protein is less solvated, its hydrophobic regions are exposed, and it is bound to the column. So now as you reduce the salt concentration, the protein will get better solvated. Its hydrophobic regions will therefore try to bury themselves within the protein, and therefore the interaction between the stationary phase and these proteins of interest will be disrupted, right? So decreasing the salt gradient is used to elute samples from the column in order of increasing hydrophobicity. More hydrophobic the proteins are, they are going to elute at lower salt concentrations. Okay. Sample elution may also be assisted by the addition of mild organic modifiers or detergents to the elution buffer. So if we take a look at it, so we have your, this is say for example the resin or the column, and we have these blue water molecules on it which are covering the resin or the hydrophobic ligand. And this is the hydrophobic region of the molecule which is also covered, and this is the hydrophilic region. So once there is interaction between these hydrophobic regions, it will bind to it, and depending upon the hydrophobicity of this hydrophobic region, the salt concentration required to promote binding will vary. For example, if your protein has low hydrophobicity, so it will bind at very high salt concentration, and when you decrease the salt concentration only gradually, it will elute out. While proteins which have higher hydrophobicity, in order to elute them, you have to decrease the salt concentration even further to promote their solvation. So coming to the advantages of HIC, large volume of sample can be loaded. Samples with high ionic strength can be used, because high ionic strength is what you will in any ways try to establish in your sample to promote this hydrophobic interaction between the column and your proteins. It is well suited to use before gel filtration, ion exchange, and affinity chromatography as a crude or preliminary step of protein purification. And sample is eluted with low salt. So the unlike in ion exchange chromatography where you have to go for desalting or dialysis, you can avoid that with HIC. Purification steps that generate large sample volumes can be coupled with this particular method. Good for samples after ammonium sulfate fractionation. Why? Because once you have done ammonium sulfate fractionation, as it is your solution has a high ionic strength, so these samples can be directly applied onto HIC without requiring any further modifications. However, do note that these techniques may require some pre-treatment of samples, that is reducing its ionic strength or increasing it depending upon the hydrophobicity of the molecule which you are trying to purify, as well as depending on the preceding purification steps. So this is what a typical chromatographic machine looks like. So other than affinity chromatography, which can be performed on those iconocolumns like I showed in one of the earlier slides, the other types of chromatography like ion exchange chromatography and size exclusion chromatography or hydrophobic interaction chromatography for their matter require sophisticated instrumentation whereby the pressure that is applied is very tightly regulated. The flow rate at which your sample will pass to the columns is also very keenly regulated. So these machines have a series of pumps. As you can see, these are the pumps that regulate the pressure and the flow rate. They have this UV cell to measure the absorbance of the solution passing through. So if you think of the chromatogram that I showed you for SEC, over there we had the MAU or the milliabsorbance unit on the y-axis. So that absorbance, which is actually a measure of the amount of protein that is present, is taken by these UV cells. And these are the different kinds of columns. So typically these long columns are your size exclusion chromatography columns, and the small ones are your ion exchange chromatography columns. And along with it, you have this connected system where you have the fractional collectors where you can collect your different fractions of protein. So that is all about protein purification, beginning from cell lysis to ammonium sulfate precipitation and centrifugation, and finally different chromatographic techniques. Now once you have your final clean protein, you simply run it on an SDS page to help visualize it to see whether it is indeed clean or whether there are any other contaminating proteins in it. And then you can use your protein for subsequent analysis, or you can add 10% glycerol to it and freeze it and store it at -80 degrees centigrade till further use. So I hope I have been able to clarify the different steps of protein purification, the different considerations to keep in mind, the different techniques, each with their individual advantages and disadvantages. And I hope that these chromatographic techniques I have been able to discuss and explain them clearly, because these techniques are very crucial and they are used commonly on a day-to-day fashion in any protein purification lab. So if you are working with protein purification, you would be for sure doing more than at least two of these chromatographic techniques. So affinity chromatography is the most common, followed by ion exchange chromatography and size exclusion chromatography. And SEC is typically coupled with affinity chromatography or with ion exchange chromatography as a final step of purification. So in case you have any doubts, do feel free to leave it in the comment section. If you enjoyed this video and if you found this useful, do like and share it and subscribe for more such content. And I will see you again next time. Bye.